Okay, so my name is Dr. Bradley Baumgarner, and this is a quick presentation of what the research that I'm going to be doing this summer. And I'm looking to recruit two students to work on this project with me. And hopefully it'll stir some interest, and I think we have a really good project for the summer. And so the title of the project is Can the environmental endocrine disruptor toluene, which we refer to simply here as the letters TF, as the abbreviation TF, can this TF compound, which is a additive in fertilizers, in marine paints to prevent fungal growth, so it's an antifungal agent. It's present in aquatic environments. It's lipid soluble, but also partially water soluble, so found especially in coastal regions. So can this antifungal agent TF negatively impact hypoxia tolerance in mammalian cardiomyocytes? And the reason this is important is because the cardiomyocytes in all mammals, but especially in relation to human cardiomyocytes, during embryonic development, they must maintain a very high capability of metabolizing glucose under low oxygen conditions. That is, they must be able to switch from an aerobic to an anaerobic glucose metabolism very readily to survive embryonic development. And so it's important to understand if this compound could negatively impact glucose tolerance, or excuse me, negatively impact the ability to switch from aerobic to anaerobic glycolysis and survive, therefore, low oxygen, i.e. hypoxic conditions during gestation. So this is some data that we have already gathered, and this is the impact of 1 micromolar and 0.5, or in this case, this is 0.5 micromolar impacts on glucose metabolism, or specifically the rate of glycolysis. And the iPSC that you see here, that stands for induced pluripotent stem cells derived that are cardiomyocyte CM. And these are human fibroblasts that we convert to pluripotent stem cells and then differentiate into fully functional cardiomyocytes. During differentiation, so on the far left, this is at day 0, the beginning of differentiation of the cardiomyocytes, and it's an approximate 18 to 20 day process. So this is at 0, 9, about halfway through differentiation, and 18 days differentiation. One thing you'll notice is that early, before the cells have differentiated into fully developed cardiomyocytes, the addition of half a micromole of this toluene compound actually increases glycolytic rate under basal, which is what I'm highlighting, and the increase that you see here in both the blue and the red being the TF treated cells and the blue the control. This is when we shut down aerobic respiration, where this arrow where it says ROTAA, that's a rotenone annamycin. That is where the cells are forced to compensate and increase glycolytic rate anaerobically. Therefore, that would dictate largely hypoxic tolerance. As you see, it increases anaerobic glycolytic rate early on at day 0 at the beginning of differentiation. No difference, although slightly less, at midway, and by the end of differentiation, these are cells that have been continuously treated with this compound for 18 days. The trend is completely reversed from day 0. As they differentiate, the cells become less tolerant to TF, and that is, it reduces glycolytic rate. And if you look at the bottom, the basal glycolytic rate are the first 3 dots, the compensatory or the anaerobic rate are the second 3 dots, data points, where it increases, and there's a compensation anaerobically because the cells are forced to compensate. So this, again, directly relates to hypoxic tolerance. So as you can see, the state of development in human cardiomyocytes makes a big difference. But the important thing is, in differentiated cells, it significantly reduces here and here. It significantly reduces both basal and compensatory glycolysis, both of which would indicate a reduced capability of surviving hypoxic conditions during gestation and early embryonic development of the heart. Another factor is the number and the density of mitochondria. So this particular slide is looking at the density of mitochondria. The mitochondria here are in rat cardiomyocytes on the left, human cardiomyocytes on the right, in a very similar trend. That is, with a 0.5 and 1 micromolar TF treatment during cellular differentiation of rat cardiomyocytes, the red mitotracker red dye stains active mitochondria. And as you can see, the density, the number of mitochondria is increasing significantly with the addition of 0.5 or 1 micromolar thiofluonide or TF. Here with the human cardiomyocytes, again, here we're treating with 0.5 micromolar TF and again, a highly significant increase in mitochondrial density. This indicates that the compound TF and is in agreement with our previous slide that it reduces the aerobic capacity and perhaps the anaerobic glycolytic rate, and the cells naturally compensate by increasing the density of mitochondria. That is, the cells are responding to reduced glycolytic capability under basal and hypoxic or compensatory conditions, as we listed on the previous slide, by increasing the number of mitochondria. That's what human cells, especially muscle cells, naturally do. They increase the density of mitochondria to become more efficient at generating ATP from a lower glycolytic rate in this case. So it's consistent with the previous slide that glycolytic rate overall, aerobic and anaerobic, is being negatively inhibited. So what I hope to do this coming summer is to examine the impact of this compound, thiofluonide compound, on the hypoxic tolerance of H9C2 rat cardiomyocytes. These differentiate in about 7 days. They're an easy model to work with. Students have a pretty easy time learning to culture these very using sterile technique. The differentiated cardiomyocytes will be exposed to hypoxia, and that is a 5% oxygen compared to 21% in normoxic conditions. And we'll do it using an environmental chamber that I already have, where we can reduce the atmospheric oxygen levels to around 5%, which is kind of the typical definition for human cells of hypoxic conditions. After cells are exposed to hypoxia, we'll analyze the glycolytic rate, as described previously on the previous slide, mitochondrial stress test, and this is essentially determining how efficient the cells are at generating or utilizing oxygen in real time to go along with the increase in mitochondrial density that we saw in the previous slide, and the amount of oxidative stress. Following hypoxia, increases in oxygen consumption, especially if they're increasing mitochondrial density, could cause a major increase in lipid and protein oxidation, which could contribute potentially to cell death. And finally, to follow up on that, we'll be analyzing cell death pathways, but particularly caspase-dependent pathways many of you have learned about in your cell biology classes, to determine if cardiomyocytes increase the rate of cell death following hypoxic stress. So I hope that this project is appealing to some of you, and if so, please feel free to contact me via email, and I hope to see you at the recruiting event as well. Thank you.